So the NADH that is produced by glycolysis is unable to cross the inner membrane of the mitochondria because the NADH is a relatively very large molecule and it is impermeable to the dense inner membrane of the mitochondria. So the electrons of NADH must be transported through the membrane in an alternative route to be utilized in the ETC to produce ATP. So that alternative route to carry the electrons is to convert to utilize those electrons and convert it into a molecule that is able to cross the membrane. And that molecule is malate. So we have oxaloacetate that is present in the inner membrane space of the mitochondria. And the oxaloacetate is able to gain the two hydrogens. So we take one of the hydrogens from NADH and the free-floating H+, and we add the hydrogens onto oxaloacetate and we get malate. And the enzyme that catalyzes this reaction is malate dehydrogenase, which is the same enzyme that is also being used in TCA, the TCA cycle, when we are converting from oxaloacetate to malate. So the two hydrogens are passed on to oxaloacetate, NADH gets oxidized into NAD+, and now we have malate. Malate, it has this gained hydrogen right here and this gained hydrogen right here, so it lost that double bond character. The malate is able to cross the membrane through a special transporter known as the malate alpha-ketoglutarate transporter. So the malate is going to cross the membrane. Once it crosses the membrane, it is going to release the two hydrogens that it gained. So NAD+ is now going to become reduced into NADH and H+, and we once again form that oxaloacetate. So we lost the two hydrogens, we reformed oxaloacetate, and it was catalyzed by the same enzyme, malate dehydrogenase, because it can be a reversible enzyme. So this oxaloacetate now has to leave the matrix of the mitochondria because we do not want a buildup of oxaloacetate. So in order for the oxaloacetate, since it cannot leave, it does not have a transporter and it is not permeable to the membrane, it must be converted into something else, just like it was on the other side, in order to leave the matrix. And the oxaloacetate is going to be converted into aspartate. It is converted into aspartate through a special enzyme known as aspartate aminotransferase. The aspartate aminotransferase is able to transfer the amino group of glutamate onto oxaloacetate. So this amino group on glutamate gets transferred to this carbonyl on oxaloacetate, and we get aspartate. So the only difference between oxaloacetate and aspartate is that we had a double bonded oxygen, but now we have an amino group. And that double bonded oxygen on oxaloacetate is transferred onto glutamate. That forms alpha-ketoglutarate. So what happened is that we had a transfer of two functional groups. The amino group was transferred to oxaloacetate, and the double bonded oxygen was transferred onto glutamate. So now we have alpha-ketoglutarate and we have aspartate. The aspartate will exit the matrix through the glutamate aspartate transporter. Once the aspartate exits through the glutamate aspartate transporter, it will be able to reform the oxaloacetate that is necessary to convert into malate. So the aspartate is converted to oxaloacetate with the same enzyme, aspartate aminotransferase. So going back to over here where we were forming alpha-ketoglutarate, the alpha-ketoglutarate is able to leave the matrix through the malate alpha-ketoglutarate transporter. So now we have aspartate and alpha-ketoglutarate on the inner membrane side. And the same thing is going to happen. We're going to have a transfer of two functional groups because it is the same enzyme, aspartate aminotransferase. So the functional group of alpha-ketoglutarate and aspartate will be transferred. Aspartate will gain a double bonded oxygen and form oxaloacetate, and alpha-ketoglutarate will gain an amino group and become glutamate. So it is the opposite of what happened on the other side. On the other side, it was a transfer between glutamate and oxaloacetate, but now on this side, it is a transfer between aspartate and alpha-ketoglutarate, catalyzed by the same enzyme, aspartate aminotransferase. Now we once again have oxaloacetate, and the oxaloacetate is ready to form into malate, and the malate is going to be ready to carry another set of electrons into the matrix. And this is the malate alpha-ketoglutarate and glutamate aspartate transporter mechanism.